Hey guys, welcome back to EduCat for NEET. In today's video, we will discuss about plant hormones. And the first plant hormone is auxin. So let's start the discussion about auxins. Talking about the biosynthetic pathway from where it is derived from tryptophan or indole. This is a kind of amino acid and auxin is derived from the amino acid which is known as tryptophan. The location where it is synthesized in the plant body is leaf primordia, in this region of the leaf, in young leaves, in developing fruits, and in growing apices of stem and roots. These are the growing apices. These roots will grow further downwards. So auxin will be synthesized here. And talking about the stem apice, auxin will also be synthesized there. Now, where it was first isolated? In the first time, it was seen in human urine. If we talk about its plant origin, then the scientist F.W. Went, he isolated this hormone auxin from the coleoptile of oat seedlings. Coleoptile is nothing but the tip of the stem. It was first discovered by Charles and Francis Darwin. They observed the first time that the coleoptiles of canary plant or the canary grass, they move towards light. Phototrophism means movement towards light. So Charles and Francis Darwin first observation they did, and they concluded that coleoptile, the area where coleoptile is present, there auxins are synthesized, and due to the synthesis of auxin, the phototrophism phenomenon is brought about. That means plant move towards light where auxin is synthesized more. And they only observed it, Charles and Francis Darwin, but F.W. Went was the first one who isolated it, means who removed it, or for observation he took it out from the coleoptile of oat seedlings. Then we have types of auxins. We have IAA, that means indole 3 acetic acid. This is present in plants, while IBA, indole butyric acid, this is present in plants. These both are natural, means they are obtained naturally. While talking about synthetic, they are created in the lab, which is NAA, naphthalene acetic acid. This is synthetic, means it is created in a laboratory. And another one, 2,4-D, which is also synthetic, 2,4-dichlorophenoxyl acetic acid. This is also synthetic. So we have got two examples of natural acetic acids, indole acetic acids, not acetic acids, auxins. So IAA and IBA are natural, they are present in plants, while NAA and 2,4-D are synthetic, they are created in the laboratory. While talking about the uses of auxins, auxins has a variety of uses. First of all, they initiate rooting in stem cuttings, plant propagation. If you cut out a root of, for example, rose plant, you can see that in a few days another rose plant will grow from it. This is nothing but vegetative propagation, in which we cut a piece of the plant. In this case, we are cutting the stem cutting, plant propagation. So this, when the root arises from the stems, this is with the help of auxin hormone. Promotes flowering in pineapple. In the pineapple plant, auxins promote flowering. When auxins are produced in large amount, then flowering takes place in pineapple. They prevent fruit and leaf drop at early stages. They prevent the fruit dropping. If the fruits drop, then we have no harvest, and leaves also drop, then the plant will die soon. So auxins prevent this fruit and leaf drop at early stages. This causes abscission of older parts like older leaves and fruits. They prevent falling of young leaves. If there are old leaves and fruits present, then they cause abscission, means they cause fall off of these fruits. They induce parthenocarpy in tomato. For example, tomato. Parthenocarpy means the growth of fruits without the help of seeds. So they induce parthenocarpy, seedless fruits. If parthenocarpy is induced in tomato, then that means you will not able to see seeds inside this tomato. Tomato is a fruit, so parthenocarpy is nothing but seedless fruits. In example tomato, auxin help in parthenocarpy. Auxin is also used as a herbicide, means the killing of herbs. If there are weeds present or any other herbs which will damage, which are not our desired herbs, then auxin can be used to make them die. Now we also talk about 2,4-D, which is a synthetic auxin which is produced in the laboratory. This kills dicot weed, does not affect monocot. So if you have all monocot plants in a field, then you can spray this 2,4-D. This auxin will kill all the dicot weeds present in that field. It will not harm the monocots, and you can enjoy a weed-free lawn. And the last use of auxin is it controls xylem differentiation. So in this way it helps in cell division also. It has a little bit role in xylem differentiation. It helps xylem to differentiate and to grow, so therefore it is helping in cell division. What is apical dominance? Now we will talk about apical dominance. Apical dominance is the growth of apical bud. It inhibits the growth of lateral buds. If, for example, this is the apic. Apic means nothing but the tip of the stem or root. In this case, we have the stem of the tip of the stem. We see a bud growing there. This bud will prevent the growth of lateral buds. If the bud is dominated on the apic, then the lateral buds cannot grow. So this is nothing but apical domination. Decapacitation means removing of shoot tips. If we cut out this tip, then you will help the lateral buds to grow. That is known as decapacitation. Application. The application is seen in tree plantation. You can see that tree plants are short in height, and this is also seen in hedge making. Decapacitation is done and apical dominance is stopped. Apical dominance also happens due to the auxin growth only. This was all about the hormone auxin. We will revise it one more. First, we learned about its biosynthetic pathway. From where did we got this? This is nothing but it is obtained from the amino acid, which is tryptophan or indole. Then where it is synthesized? Where is auxin synthesized? It is synthesized in the leaf primordia or the young leaves or the developing fruits, and also in the growing apices of stems or roots. Where was it discovered? It was first discovered by Charles and Francis Darwin. He observed that the coleoptile of canary grass, it moved towards the light due to auxin synthesis. Auxin was synthesized more on this part, so this stem, this canary grass, moved towards the light, which means it exhibited phototropism. But for the first time, it was isolated by F.W. Wendt from the coleoptile of oat seedlings. We also observed some trace amount of auxin in the human urine also. Then there are types of auxins. Auxins can be either natural or they can be synthetic. We have seen two examples of natural auxins, that is IAA and IBA. They are naturally present in plants, while synthetic, they are created in the laboratory, that is NAA and 2,4-D. We have seen the various uses of auxins, which include they initiate the rooting, they initiate the coming of roots in the stems cutting, therefore they help in plant propagation. They promote flowering in pineapples. They prevent fruit and leaf drop at early stages. Young fruits and leaves are, they prevent them from falling, while they cause abscission, they cause fall off of older parts such as leaves and fruits. They induce parthenocarpy, which means seedless fruits in tomato, and they can also be used as a herbicide. Herbicide means killing of herbs. We have also seen that 2,4-D, which is a synthetic auxin. This helps in the killing of dicot weeds in a monocot field, and it have no effect on the monocots. Therefore, we can enjoy a weed-free lawn. The last but not the least use of auxin is it helps in control of xylem differentiation. In this way, it helps in cell division. What is apical dominance? Apical dominance is growth of apical bud, and this apical bud inhibits the growth of lateral buds. When decapacitation is performed, means if we cut the apical bud, then removal of shoot tips. If we remove these tips, then we can see the growth of lateral buds. This application is seen in tree plantation and hedge making. This was a brief summary about the auxin hormone. If you enjoyed this video, like, share it with your friends, and subscribe. If you have any doubt, you can comment down below. And thank you for watching.